Finally, the sample have reached the sample conditioning panel. Sample conditioning panels are the final stage to handle process samples, which are very important to ensure instruments or gas analyzers works properly. Its target is to provide process sample filtration, temperature control, pressure control, fast loop and sample flow adjustment, multi-stream switching, calibration, introduction to analyzer and lab sample facility. Sample conditioning panel are located at analyzer shelters or stand alone in the field. In this panel there are different components, and each component has a different function. We will start discuss the filtration function of the sample conditioning panel. Sample filtration. Contaminants in the plant samples are reported to be the most frequent cause of problems with online analyzers. The filter must be able to separate efficiently a non-continuous phase contaminant from the continuous sample stream phase. In addition to filtering, a good sample filter should have this characteristics. The filter must not change the composition of the sample. Capable of operating for a reasonable period between scheduled maintenance checks, it should not be vulnerable to unscheduled problems, such as filter element plugging or rupturing, between regular maintenance checks. Filter should be designed for easy and uncomplicated maintenance. Filter elements should be rugged and not susceptible to handling damage. Also, the unassembled housing should have a minimum number of loose parts. The housing should be designed so that it is virtually impossible to install a filter element incorrectly. A filter should introduce minimum lag time into the system. Since large reservoir volume is desirable in many filter applications such as compressed air or water filters, filters not specifically designed for sampling usually are not suited for analyzers. Some filters are designed to make the following separations, in addition to removing solid particles. For gas samples, filters could be used to remove liquid droplets. And for liquid samples, filters could be used to remove immiscible liquid droplets and gas bubbles. Let us talk more about separating liquids from gases, or as we call it, the coalescing process. The filter is designed to perform two functions. The first one is the filtration, where solid particulates are removed from the fluid stream by the filter. The second function is the liquid droplet separation. Glass microfiber filter tubes efficiently separate suspended liquids from gases. The filter fibers capture the fine liquid droplets suspended in the gas and cause the droplets to run together to form large drops within the depth of the filter tube. The large drops are then forced by the gas flow to the downstream surface of the filter tube, from which the liquid drains by gravity. This process is called coalescing. Since the coalesced liquid drains from the tubes at the same rate that liquid droplets enter the tubes have an unlimited life when coalescing liquids from relatively clean gases, and the filters operate at their initial retention efficiency even when wet with liquid. The flow direction is inside to outside to permit the liquid to drip from the outside of the filter to the housing drain. Also, for liquid samples, filters can be used to remove suspended gas bubbles from liquid, eliminating the need for deaeration tanks, baffles or other separation devices. Flow direction through the filter is outside to inside, and the separated gas bubbles rise to the top of the housing and are vented through the drain port. If slipstream sampling is used, the separated bubbles are swept out of the housing with the bypassed liquid. Filter tubes rated at 25 micron are a good choice for gas bubble separation. Let us talk more about slipstream or bypass sampling. It is essential to minimize lag time in the sample system. Ideally, a filter should be located at the point where the low-flow stream is withdrawn to the analyzer. This arrangement permits the main volume of the filter to be swept continuously by the high flow rate system, thus minimizing lag time. At the same time, only the low-flow stream to the analyzer is filtered, thus maximizing filter life. If bubble removal from a liquid is a requirement, this function may be combined with slipstream filtration, since the recommended flow direction for bubble removal is outside to inside, and the separated bubbles will be swept out of the housing by the bypass stream. In this case, the liquid feed should enter at the bottom of the housing and the bypass liquid exit at the top of the housing. To better understand the difference between the particle filtration and the coalescing filtration, let us check this comparison. First, the orientation. In the particulate filtration, orientation of the filter is not critical for proper operation, while in coalescing filter, orientation of the filter housing is critical for proper operation. The drain port must face down to allow liquid to drain from the drain port. The second point is the input port and the direction of flow in the filter element. In the particulate filtration, gas stream enters top port and moves through a filter element from outside to inside. While in coalescing filter, gas stream enters side port and moves through filter element from inside to outside. Third is the function of the filter element. In the particulate filtration, filter element captures any particulate suspended in gas, only a small volume of gas passes through the element. While in coalescing filter, the filter element captures fine liquid droplets suspended in the gas, forcing them to coalesce or form large drops within the filter element. The fourth point is the exit of the gas from the elements. In the particulate filtration, most of the gas bypasses the filter element and exit through the bottom port and return to the process line. While in coalescing filter, all the gas passes through the filter element. And dry filtered gas exits through the top port and flows to process analyzer, and liquid exits out from the drain port. Slipstreaming plus coalescing. Slipstreaming plus coalescing requires two stages of filtration. The second, coalescing stage, must be in the sample line to the analyzer and should be as small as possible to minimize lag time. Now we will see different filters designs and shapes from Swagelok Company. We can see here the filter mounting brackets, the inlet and outlet ports, the nut that is used to open the filter housing and clean or replace the filter element. We can also see the filter element itself and the perforated sleeve that provide support for the filter element. In this slide we can see three different shapes or types of the filter. The first one, the filter element is welded in the filter body, so if contaminated or blocked, the complete filter with its body is replaced. The second one is used as an inline filter just before the analyzer, and it serves as the final filtering stage for the sample before it enters the analyzer. Note that the filter element can be removed for cleaning or replacement without changing the filter body. The third filter design can be used as inline or a bypass filter element because it has two outlet ports. Also, the filter element can be removed separately from the filter body.